Hi, hello, my name is Beatriz Villaseñor-Altamirano. I'm a PhD student from the International Laboratory of Human Genome at UNAM, Mexico. My PI is Alejandro Medina Rivera, and I'm presenting our work entitled Transcriptomic Meta-Analysis of Chronic Obstructive Pulmonary Disease using PulmonDB. Chronic obstructive pulmonary disease, or COPD, is an umbrella term for emphysema and chronic bronchitis, which lead to breathing capacity in patients that have this disease. Across all of these years, different researchers have analyzed COPD using high-throughput data with microarrays and RNA-seq. However, the results change across different publications. Therefore, we aim to have a consensus transcriptomic long profile in COPD patients using different studies that are in PulmonDB with a meta-analysis. PulmonDB is a gene expression database with public experiments that we developed. This data has been consistently normalized and manually curated to obtain a consistent ontology annotation. PulmonDB uses sample contrast that are the difference between a test and a reference sample from the same experiment. Each reference is randomly selected from the control group per experiment. So using PulmonDB, we selected experiments from lung tissue and those that contrasted COPD versus control samples. Additionally, we use raw data from GEO to compare the differential expression results, and then we perform a meta-analysis using a random effect model. As a result, we first present the table showing 7 experiments that we are going to use in this meta-analysis. As you can see, the number of samples, platforms, and years are different, but all has a normal category and group of COPD patients. Then we analyze the data looking for batch effect, plotting the data in a PCA. We found that in both cases, the principal components are driven by the experiment ID, which are shown in different colors. We also see the same effect using unsupervised clustering, as you may see in figure 1b. But in pre-processed raw data, is more predominant this batch effect than in PulmonDB data. Before performing the meta-analysis, we compare the results of differential expression analysis using PulmonDB and pre-processed raw data per experiment. In figure 2, the x-axis is the log-fold change results of PulmonDB, and the y-axis is the log-fold change of pre-processed raw data. Each dot is a gene, and each plot corresponds to the different results that we obtain from all of these 7 experiments we selected for this analysis. All 7 experiments show a good correlation across results. In other words, genes upregulated in PulmonDB are also upregulated in pre-processed raw data, and genes downregulated in PulmonDB are also downregulated in raw data, showing that PulmonDB results are consistent with pre-processed raw data. Finally, we performed the meta-analysis using a random effect model per gene. Using PulmonDB data, the p-values were corrected for multiple testing, and those with a value less than 0.01 were plotted in a heat map showed in figure 3a. Rows are genes, and columns are experiments. An example of one upregulated gene is the CXCL12, or CXC motif chemokine ligand 12, that is upregulated in all 7 experiments. Another one is annexin A3, or ANX-A3, that is downregulated. In conclusion, PulmonDB data correlates with pre-processed raw data, and we were able to obtain a robust profile for COPD samples in lung tissue using PulmonDB. Thanks to all the people that helped me in this analysis, my collaborators, Patrick, Alejandro, my lab, my PI, and institution. Thank you for listening, and I will be happy to answer any questions.